Hello everyone, we are the group 4 and today we will be discussing gene therapy. What it is, how it works, and a major scientific breakthrough that demonstrated its potential. We will also look at the historical origins of gene therapy, why it is considered both promising and challenging. So many humans' diseases are caused by defective genes, so these conditions cannot be cured by traditional medicines, so gene therapy offers a potential method to treat or cure genetic disorders. So many diseases originates from mutations or defects in person's DNA. Traditional medicine usually treats symptoms but do not fix the defective gene itself. Gene therapy, however, targets the root cause by attempting to correct or replace the faulty gene instruction. Gene therapy works by inserting healthy genetic material into a patient's cells. This new DNA can compensate for or correct the defective gene. Once the correct instructions are present, cells may begin functioning normally, providing long-term or even permanent improvement. In 2015, researchers achieved a stunning breakthrough. They restored the hearing mice that were deaf due to a genetic mutation. Even more impressive, hearing improved to 25 decibels, which is as soft as a whisper. This showed that gene therapy could meaningfully restore function.
So, good evening, everyone. Let's continue the report of Giancarlo Banglos, which is the introduction of gene therapy. The researcher or the scientist discovered an ANC. So, successful was possible because scientists used a powerful viral vector called ANC. So, what is ANC? ANC, or the ancestral adeno-associated virus, which was discovered in 2015 in the lab of Dr. Luke H. Vandenberg at the Massage Eye and Ear. This is used for developing a gene therapies to treat rare and ultra-rare retinal disease. So, ANC, the vector can reach the outer hair cells, tiny sensory cells deep inside the cochlea, responsible for detecting sounds vibration because these cells are normally difficult to access. ANC mark a major advance in gene delivery about technology. So researchers used a improved gene therapy vector, which is the ANC, developed at mustache, eye, and ear, enable delivery of genes to hard to reach outer hair cells in the gut. So, the history basis of a gene therapy, a concept of human gene therapy first emerged in 1971, inspired by early recombinant DNA experiments, involves inserting foreign DNA into tissues to eliminate genetic disease. So, what are the promises and challenges of the scientists to identify the gene therapy or to find a solution for a gene therapy? uh, the promises and challenges are gene therapy is highly promising in treating genetic illnesses, considered one of the most innovative but also challenging medical fields, offers potential cures but involves risk and ethical concepts. So, promise and challenges. Gene therapy is highly promising in treating genetic illnesses. Considered one of the most innovative but also challenging medical fields, offers a potential cures but involves risk and ethical concern. So that gene therapy represents a powerful advanced approach to treating diseases at their genetic roots. The 2015 study proved it is real-world potential, showing that even sensory functions like hearing can be restored. As the field continues to grow, it offers hope for many future cures, provided we proceed with responsibility and ethical care.
Good day, everyone. So, to continue the discussion, let's talk about the basic processes of gene therapy. The basic process. So, when we talk about basic process. It is a fundamental and systematic process that helps us to have a smooth and well organized preparation in dealing with gene therapy. Also, in order to have an outcome that is fit to what is really the significance of having gene therapy in a certain disease or disorder. So let us so let us try to understand or let us know what is gene therapy. So, gene therapy is a biomedical technique that aims to treat or prevent disease by modifying genetic material inside a patient's cells. So, in other words, it is a treatment by modifying the genetic material of patient's cell. Yet, this kind of therapy is applicable to those genetic disorders like the leukemia. Because lots of people that have leukemia is genetically because it talk about a blood disease, and by the help of CAR T cell therapy, the patient's body or specifically the immune system of the patients has a chance to fight the leukemia. Why is it? Why is this so? It is because when when the patient undergo a CAR T cell therapy, the T cells of the patient is removed, genetically modified to better detect and kill cancer cells, and then return to the body. That's why some leukemia cases is advisable to undergo a CAR T cell therapy because this help the body kapitang um, mas malesen or mas kanang dili kayo mas malesen ang malfunction situation, and para po dili bitaw murag bulay na lang ng tawon ang lawas sa patient. So that is gene therapy. Now, according to Whistler 2017, there are basic approaches in dealing gene therapy, and it includes, so first is replacing a mutated gene. So it is a defective or disease-causing gene is substituted with a healthy functional copy. So this approach is commonly explored for inherited genetic disorders where the mutation results in loss of function. So loss of function, meaning wala na nag-function normally. And if wala na nag-function normally, wala na yung barrier sa katong mga cancer cells. Which means, scatter na sila and possible maapektuhan ang other healthy cells. So in this approach, the damaged and the non-working gene is removed and replaced with healthy version so that the cells can function normally again in order to keep fight of the cancer cells para dili mas mogabi ang disease o kanang mas maagapan siya. Next is activating a faulty gene. A malfunctioning gene that produce harmful effects is switched off or silent. These techniques like RNA interference or RNA or gene editing tools, example is CRISPR-Cas9, can be used to disable harmful gene activity. So in this approach, ang mga therapy ani is ilahang i-turn off ang mga harmful gene. Why? Para dili nakaproduce of proteins. It is because when makaproduce sila og proteins, magkakos dayon og disease. So kung dili sila ang baton, kung dili pasagdara sila, din mag-produce sila protein to the point nga mugabi na kaayo ang disease. Lisod na dayon siya i-prevent. Impossible, ma-appeal ang other healthy cells to the point nga pila na lang dayon ka-person sa body ang functionally normal. So that is the second approaches. And last is introducing a new gene. A therapeutic gene is added to help the body fight or prevent a disease. So this is often used for conditions where supplying an additional gene can restore normal cellular functions or stimulate immune response. Example is the cancer gene therapies. So, in this approach, ang mga therapy ani is mag-give sila og brand new. Mag-add sila og mga gene or cells dito sa old one para help to fight back the cancer cells. So, kani siya is mostly applicable ni siya sa mga cancers. It is because, di ba, ang cancers is needed siya of new cells to help fight the cancer cells. But take note, na dili ka sa ara ang session ani nga therapy. It is because paglala na kaayo ang disease, na siya sa mga stage 3 or stage 4, lisod na kaayo pildihon ang cancer cells. Mao dili siya kaya in one session. So that's the basic approaches of gene therapy. So, understanding the basic processes of gene therapy is important because it shows how diseases can be treated at their genetic source. These approaches guide the development of target, long-lasting treatments, and open new possibilities for managing genetic disorders and other serious diseases. In short, basic processes of gene therapy is very crucial and important. Why? It is because it helps to treat, prevent, and manage the genetic diseases. Makahatag siya para dili, makatabang siya rather, para dili murag gulay ang pasyente, para dili magsuffer o maayo sa pain ang patient. Namang way uban therapy nga dili jud mapildi ang disease, but at least you try na prevent and you are not wasting your last days for nothing. That each day you are still fighting. And also, helpful siya in managing the genetic disorders. Kaya naamang good pan disorders nga if makatry og therapy, din dili na makontinue, ma-triggered ang cancer cells. Dara mas mo spread out natin to the point nga maapektuhan na halos tanang part sa body. So remember, basic processes of gene therapy is very, very important. Why? First, ilang kwaon ang harmful gene, ilisan, adan, to be back in a normal function and to help fight the disease. That's all. Thank you.
Now that we've learned about the basic processes of gene therapy, such as replacing, inactivating, or adding genes, I will explain how these therapeutic genes are actually delivered into the patient's cells. So as you can see on the screen, we have 6 bullet points that are simplified explanation of how genes are delivered in gene therapy. So we have genes cannot be inserted directly into human cells. Why? Because one important thing to understand is that gene cannot simply be placed directly into a human cell. Why? Because our cells are protected and DNA cannot enter them on its own. Because of this, scientists need a delivery tool called a vector. So again, what is a vector? So a vector is like a vehicle that carries the therapeutic or healthy gene into the target cells. So the most common type of vectors used today are, you might be shocked, but it is the one or the common used vector today, which is the viruses. This might sound surprising because when we heard the word virus, it means an infection, right? But in gene therapy, this is the common use vectors because viruses are actually very useful in medicine. So why? Because in nature, viruses are expert at entering cells and delivering their own genetic material. So scientists take advantage of their natural or of this natural ability, but in a safe way. So before using virus, researchers make sure to remove all the virus harmful genes so it can no longer cause disease. After that, they insert a healthy copy of a human gene that the patient needs. In other words, the virus is reprogrammed so that instead of causing infection, it now delivers a therapeutic DNA. Once the modified virus enters the patient's cells, it releases a healthy gene, so allowing the cell to use to reproduce a normal and functional repeat. This process helps correct the underlying genetic problem or support the body in fighting disease. So, so because of their efficiency and natural behavior, viral vectors are the most widely used and effective methods for gene delivery in gene therapy today. Without this tool, which is the vectors, the therapeutic genes would have no way of reaching the cells that needs them. And that's it. That is how genes are safely and effectively delivered inside the body during gene therapy. Again, in recap, um, uh, gene cannot be inserted directly into human cells. A vector is needed to carry the therapeutic gene into the target cells. Viruses are the most common used vectors, and these viruses are genetically modified so they cannot cause disease, and they are engineered to carry healthy human DNA instead of their harmful genes. And because viruses are naturally delivered genetic materials to the cells, scientists use this ability to transport the correct gene state. And that's all. Thank you.
Now let's talk about the two types of gene therapy, which are the somatic gene therapy and the germline gene therapy. But first, let's talk about the somatic gene therapy. So a somatic gene therapy, it targets somatic or non-reproductive cells. So this type of gene therapy works on our regular body cells, not sperm or egg cells. So we only fix the cells that are causing the illness in the person. Also, it alters DNA in affected tissues only. So the treatment changes the DNA only in the parts of the body that are sick. So for example, if the lungs are affected, we put the new gene into the lung cells. So other parts of the body stays the same. And then it benefits the patient, but changes are not passed on to the offspring, which means that the changes stay in the patient and don't get passed to their children. This is because the therapy does not touch the reproductive cells, so only the person receiving the treatment gets the benefit. So this type of therapy is safer and widely accepted ethically. So it is considered safer because it affects only one person and doesn't change future generations. So people also see it more acceptable because it is used to treat diseases, not to change traits in unborn children. So how this somatic gene therapy helps with certain diseases? So in hemophilia, we add a working gene that helps the body make the missing blood clotting factor. So this helps stop bleeding problems. So in cystic fibrosis, we give the lung cells a correct version of CFTR gene so they can make a normal mucus. So this helps the patient breathe better. So in certain cancers, we can change a patient's cells so they can fight cancer better or make cancer cells die. So for example, in some treatments, we modify immune cells so they can attack the cancer more strongly. To make it short, somatic gene therapy means the treatment helps the person who gets it, but the changes stops with them. Their children will not inherit the fix.
Good day, everyone. So today I will be discussing the second type of gene therapy, which is the germline gene therapy. So what is the germline gene therapy? Germline gene therapy involves modifying the DNA of reproductive cells such as the sperms, the eggs, or the early embryos. What makes it different from the somatic gene therapy is that any changes made in these reproductive cells becomes heritable, meaning the modification can be passed on to future generations because these changes affect not only the individual but their entire lineage. Germline gene therapy is considered highly controversial and is prohibited in many countries. So why is it controversial? It is because this type of therapy is controversial for several reasons. First, the changes it makes are permanent and can be inherited. This means even a small mistake can continue to appear in future generations. Second, there are risks of unintended side effects, such as new mutations that we may not fully understand yet. Third, the long-term consequences remains unknown, so scientists cannot guarantee its safety. And lastly, there is an ethical concern, which is the future generations who will inherit these genetic changes cannot give their consent, yet the modification will affect their entire life. So how does it work? It normally begins by targeting the germ cells, which are the sperm or egg, or it can target a fertilized egg called a zygote. Next, the DNA is modified. Scientists may insert a new gene, remove a defective one, or make precise edits using gene editing tools. Once the modification is made, the embryo grows with this edited DNA. As a result, the change appears in every cell of the developing individual. Because the reproductive cells also carries the edited DNA, the modification is automatically passed on to their future offspring. Its potential benefits are, despite the controversies, germline gene therapy also offers potential benefits. It could completely eliminate certain hereditary diseases from a family line, providing long-term solutions for conditions like cystic fibrosis, sickle cell diseases, or other inherited disorders. This means that genetic problems can be prevented even before the child is born, offering a healthier future not just for an individual but for future generations to come. That is all for my part. Thank you.
Good day, everyone. Today I'll be reporting about stem cell gene therapy. So, this topic combines two important areas in modern medicine, which are stem cells and gene therapy. So, together they provide new ways to repair damaged tissues and even correct genetic disorders. So, before we understand stem cell gene therapy, we need to first understand what are stem cells. So, what are stem cells? Stem cells are mother cells that can become any cell type in the body. So, stem cells are often called as mother cells because they are the original source of many other cell types in our body. So, uh, unlike normal cells who have only one designated job, stem cells have two abilities. Alright, so first is they can self renew, meaning they can keep on multiplying, so they can make more stem cells. Like, for example, our body needs thousands of blood cells every second. So, stem cells keeps on multiply, keeps on making more stem cells, and help supply that. Second is they can turn into different types of cells. And actually, that's what makes them special and powerful because they're very flexible. Like, for example, if our body needs blood cells, they can turn into blood cells. If our body needs muscle cells, they can turn into muscle cells. And again, the main feature of or main ability of stem cell is the ability to turn into multiple cell types. Examples are blood forming stem cells that developed into red blood cells, neural stem cells that develop into neurons, muscle stem cells that develop into muscle fibers. So, why do stem cells matter? Why do scientists talk about them so much? It's because stem cells have the potential to repair, replace, and even regrow damaged tissues. So, first is they repair or replace damaged tissues. Like, for example, if a person's bone marrow is damaged due to cancer treatment, stem cells can be transplanted to rebuild their blood system. Second is they are used in gene therapy to correct genetic diseases. So gene therapy is when a doctor fixes a broken or mutated gene. So stem cells are good carriers for this because once their gene is corrected, the healthy cells they produce will also carry that corrected gene. So third is they are valuable for medical research and regenerative medicine. uh, Scientists use stem cells to study how diseases develop and how to create better treatments for these diseases. Examples are when treating blood disorders like leukemia through stem cell transplant. Another example is attempting gene correction for sickle cell disease. Now, where do stem cells come from? There are two main types or two sources of stem cells. First is embryonic stem cell, and second is somatic stem cell. So, first, let's focus on embryonic stem cell. So, what are embryonic stem cells? So, they are taken from a 4 to 5 day old embryo or blastocyst stage. They usually come from extra embryos created in IVF clinics. So, they have a pluripotent ability. Pluripotent ability means they can become almost any cell type. Okay, so we have here examples on how they are used. So first is they can be used to grow heart cells for heart disease research. So scientists grows heart cells from embryonic stem cells for them to study heart diseases and test new medicines. Second is they can form pancreatic cells for diabetes research. So, embryonic stem cells are used in diabetes research, especially in those studies that requires or needs insulin producing cells. And lastly, they are used to study early human development. So, scientists use embryonic stem cells to help them understand how organs are formed, why some birth defects happen, and how early human development works.
Hello everyone. Today I will be discussing somatic stem cells, including what they are, where we can find them, how they work, their limitation, and their importance in the human body. So what are somatic stem cells? Somatic stem cells, also known as adult stem cells, are special cells found in our bodies after birth. Their main job is to repair, replace, and maintain damaged or old cells, unlike embryonic stem cells that can turn into almost any type of cells. Somatic stem cells are usually limited to becoming the type of cells that belongs to the tissue where they came from. So where we can find them? So somatic stem cells can be found in many different parts of the body. They are located in the bone marrow, brain, skin, liver, blood vessels, and even in the muscles. Each tissue has its own type of somatic stem cells that helps repair damage in the specific area. So how do somatic stem cells work? So most of the time, somatic stem cells stay inactive. They only become active when the body needs them. For example, when we get injured or when cells die, once they are activated, they start dividing to produce new cells. These new cells then develop into the specific type of cells that is needed, such as blood cells, skin cells, or liver cells. This process helps the body heal and recover from damage. So what are the limitations of somatic stem cells? Although somatic stem cells are very useful, they also have limitation. They can only turn into certain types of cells, usually the ones from the original tissue. For example, a skin stem cells cannot become a nerve cell. They also exist in small numbers in the body, which makes them harder to collect, and compared to embryonic stem cells, they divide more slowly and are less flexible. So what are the importance of somatic stem cells? Despite this limitation, somatic stem cells are extremely important. They are responsible for repairing tissues, healing wounds, and keeping our organs functioning normally. They are also used in important medical treatments like bone marrow transplants, which help patients with serious blood diseases. Somatic stem cells are also being studied for future regenerative therapies that may one day help treat damaged organs. In summary, somatic stem cells are the body's natural repair system. They are found in many tissues, they become active when damage occurs, and they help the body heal and stay healthy. They may have limitations, but their importance in medicine and healing is undeniable. Thank you.
Good day, everyone. So I'm Mayan Dalman. So now let's talk about the bioethics gene therapy. So there are ethical issues involved in gene therapy. Some of the inquiries cited are: question one, how can good or bad uses of gene therapy be distinguished? So good uses of gene therapy are those that help people who are sick, such as treating or preventing serious genetic diseases, while bad uses happens when people use gene therapy for things that are not related to health, like choosing a baby's looks or trying to make someone smarter. And then question two, who decides which traits are normal and which are a disability or a disorder? So there is no single person who decides what traits are normal. Medical expert, ethicists, and society all have a role in making these decisions. And different cultures may see things differently, so it's important to avoid judging or discriminating against people with certain traits. And then question three, will high cost make gene therapy available only to the world? So right now, gene therapy is very expensive. Because of this, only people who can afford it may get access to the treatment. So this creates a problem of inequality because not everyone will be able to receive the same level of health care.
Hello everyone, I am Juliet and today we'll be answering few questions regarding gene therapy. So here is the first question. Could widespread gene therapy reduce acceptance of human differences? The answer would be yes. Yes, widespread gene therapy could make people less accepting of human differences. Some might start believing that certain traits or disabilities should be changed, which can lead to less respect for people's natural variations. Now let's move on to the second question, or should I say the last question for my part. Here is the question. Should people be allowed to use gene therapy for enhancement like height, intelligence, or athletic ability? The answer would be no. People should not use gene therapy for enhancement. You know why? Because it can cause unfair advantages and it makes society more unequal. And it put pressure to people to try to be perfect. And always remember, gene therapy should be used for treating diseases, not for boosting abilities. You have any question? Did you get it? Alright, you don't have any question? Thank you so much for listening, everybody.
Good day, my name is Michaela Disipida and I'm going to discuss about the germline therapy. So what is germline therapy? Germline therapy is a therapy where it involves modification of genes or DNA in reproductive cells. When we say reproductive cells, these are the egg cells, sperm cells, or even embryos to prevent hereditary diseases that can be passed to all future generations. So the example of this disease is cystic fibrosis. So it offers the prospect of eliminating genetic disorders from the entire family lineage. But this therapy is currently banned or been restricted by other countries such as United States for their safety, ethical, and societal concerns. So we have here the effects of gene alteration on the unborn child and future generations. So first, we have intended health benefits. So it can prevent the transmission of inherited genetic diseases. So it may improve the child's long-term health by correcting harmful mutations. So there is a potential to eradicate diseases through germline therapy because it offers the prospect of completely eliminating specific genetic disorders such as cystic fibrosis or Huntington's disease from a family line and a human gene pool, as the correction is heritable. So now let's proceed to number 2. So germline therapy has this uncertain long-term effect. So we have the long-term effects, but uncertainty. Why is that it is uncertain? So it is because altering reproductive cells creates heritable changes that can affect not just the individual or the target individual, but also to the off-target individual. So, irreversible off-target genetic changes and the fact that a gene that is harmful in one environment can be beneficial to the other, making the long-term consequences of any modification impossible to fully predict or control. So, dili siya makontrol because pwede siya beneficial sa isa but also harmful siya sa musunod. Dayon, this current technology man can't guarantee that the genetic modification will only affect the intended gene. So off-target effects could cause new unforeseen health problems in the individual and their descendants. So sa mga musunod pa. So it is dili siya makontrol. So it is impossible to control. So that's why alterations may cause unexpected genetic changes that appear later in life because future generations may inherit, it can be inherit, the unknown risk or complications. So now let's proceed to number 3. So germline therapy has an irreversible impact. Why is that? It is because the genetic changes are made in reproductive cells, such as sperm, egg cells, and even embryos. So these cells are heritable, meaning they are passed down to all subsequent generations. So there is a permanent alters to the human gene pool, that family line, and potentially humanity as whole. So meaning, maapektuhan ang family line or the future generations. So there is this fundamental reason. So the fundamental reason for the irreversible impact is that the DNA is modified in the cells that create offspring. So every cell in the resulting individual will carry the modification. And they, in turn, will pass it on their children, grandchildren, and so on. So the future generations. So once a change is introduced in the germline, it is difficult or it is impossible to reverse the effect in all future individuals who inherit the modification. So walay consent gikan sa mga future generations. So this is very alarming because dili lang maapektuhan ang previous individual or target individual para kuno dili maka-attain o ginana nga disorder, but also maapektuhan ang future generations because ang mga off-target individuals kay naay mga abnormalities sa ila because dili sila apin and it is irreversible impact. So mistakes or unintended mutations cannot be easily corrected across generations. So maapektuhan ang mga future generations. So now let's proceed to the fourth one or the last one, which says here that germline therapy raises ethical and safety concerns because it can create heritable genetic changes in future generations, carrying unknown long-term consequences and the risk of off-target mutations. So there are also worries about using technology for non-medical enhancements, potentially leading to social inequality or designer babies, which could involve issues of fairness and human dignity. So, consent is another major ethical issue, as future generations cannot consent to the alteration made to their genome. So, murag wala sila consent bitaw sa mga future generations, since ang mga future generations can be affected also. So, the safety concerns, there is unforeseen consequences because it's a new field. There are risks of unknown long-term side effects on the individual and their descendants. So, na mga long-term effects sa ilahang descendants, sa ilahang future generations. So, ang mga off-target effects, genetic changes could have unintended effects on other genes, leading to unpredictable health problems. So, sa ilahang genes manahimutang ang kanang therapy. So, there is this unpredictable health problems, not just the individual itself, but also to the future generations. So, sa ethical concerns, we have these hereditary changes. So, alterations made to germline cells are permanent and will be passed down to all future generations, which may see as a serious and irreversible steps. So, inherit man siya. So, it is heritable. So, it says here that it can be passed down to future generations. So, there is this ethical concerns that alterations and the product of that alteration is irreversible. The effects of that alteration, gene alteration, is irreversible. So, sa ethical concerns also nag-mention diri ang consent. So, individuals who will be affected by the therapy in the future that are not yet born and therefore cannot consent to the treatment. So, wala sila alamag because for future generations man sila, but affected sila sa cell nga therapy sa process. So, future generations cannot consent to genetic changes affecting them. So, may influence societal views that is acceptable or ideal truths. So, matawag sila mga designer babies because there are these changes sa ilahan. It's not normal. There are this effect of the therapy that is not normal for the future generations because, as what I've said earlier, na it can be beneficial to that one person but harmful to the other, especially for the future generations that has an irreversible effect to them. So that would be all. Thank you.
So, good day everyone. In overall terms, gene therapy is presented as a potential solution for treating and curing genetic disorders. So, it involves two main types. First is somatic gene therapy, which directly targeted the patient's cells. Second is the germline gene therapy, which modified germ cells that can affect future generations. So, it addresses several ethical concerns related to gene therapy, particularly regarding the authority to decide which human traits should be altered and the potential for discriminations against individuals who cannot access the therapy. So that's would be all. Thank you.